So next we're going to talk about the different routes of administration of different vaccines. So the route of administration is really important because it's the path by which a vaccine is brought into contact with the body, and this is really important for the success of the immunization. So a substance must be transported from the site of entry to the part of the body where its action is desired to take place. And each vaccine has a recommended administration route and site, and you can find this information in the manufacturer's package insert for each vaccine. And it's really important to follow this instruction, as deviation from the recommended route may reduce vaccine efficacy or increase local side effects or local adverse reactions. And just an important point that whatever route the vaccine's being given, hand hygiene is really important before administering. So the first route we'll talk about is oral. So there aren't as many oral vaccines, but this can be easier in a lot of cases as it eliminates the need for a needle and syringe. So some examples are the oral polio vaccine, which we'll talk about later, and the rotavirus vaccine. Then you have the intramuscular injection route, and this administers the vaccine into the muscle mass. So any vaccines containing adjuvants should be injected intramuscularly to reduce adverse local side effects. So an adjuvant is a substance which improves the immune response of the vaccine. So some vaccines contain these adjuvants. So any vaccines containing adjuvant should be injected IM. Then you have the subcutaneous route, and this administers the vaccine into the subcutaneous layer above the muscle and below the skin. And you can see some examples there of measles and yellow fever. Then the last route is the intradermal injection, and this administers the vaccine in the topmost layer of the skin. And the BCG vaccination is the only vaccine with this route of administration. So intradermal injection of BCG vaccine reduces the risk of neurovascular injury. And health workers say that the BCG is the most difficult vaccine to administer due to the small size of newborns' arms. And a short, narrow needle is needed for the BCG vaccine, whereas all other vaccines are given with a longer, wider needle. So this slide just shows a little bit more about the actions of vaccines. So first of all, you get recognition of the viral antigen by B lymphocytes and generation of antibodies. And you get binding of antibodies to target site on the virus surface. And this blocks interaction with the receptor and aggregation of virus particles. And then you get the complement-mediated lysis of free virus and infected cells triggered by antibodies. And you get what's called the opsonization of virus particles by antibodies. So that's the marking of the virus particles by antibodies, marking them for phagocytosis. Okay, so this is when the virus becomes engulfed by phagocytes. And then you've also got the cellular cytotoxicity of the infected cells. So this slide looks at vaccine-induced IgG and IgA, which are antibodies we touched upon earlier. So if we start with IgA, so if you remember, IgA is mainly present in saliva, tears, breast milk, and mucus secretions of the GI, respiratory, and genitourinary tracts. And these body fluids coat and protect the extensive mucosa. So it shouldn't be a surprise that IgA is heavily involved in mucosal immunity. So mucosal immunity is an area under a lot of investigation, since it has the potential to stop a virus from even entering the body, since many infections are initiated at mucosal sites. For example, nasal, oral, ocular, rectal, vaginal. And as we said, immunity at the mucosal surface involves secretory IgA, as well as low levels of infiltrating antibodies and cells from the blood. Now, live vaccines are able to stimulate mucosal immunity, whereas the inactivated vaccines are less able to do so. And it is also possible to stimulate mucosal immunity directly by inoculation of vaccine onto almost any mucosal surface, including the oral, respiratory, and genital areas. And there's good evidence that mucosal immunity may be required for optimal protection against viruses that infect via such routes. So therefore, a number of mucosal vaccines have been developed, and mucosal vaccination involves the administration of vaccines at one or more mucosal sites, leading to induction of immune responses at the mucosal site of administration, other mucosal sites, and or systemically. And the advantages of mucosal vaccination include early control of infection, effective control of non-invasive infections, and the removal of the need for injection. However, the disadvantages are, in practice, it is difficult to induce an effective IgA response, and there can be antigen instability at mucosal sites. So some of the examples of mucosal vaccines that have been designed are the nasal influenza vaccine for children, the oral polio vaccine, and the oral rotavirus vaccine. So there's some examples of mucosal vaccines. So IgG, on the other hand, is the main antibody in the blood and is required for viral clearance in the blood. So although IgA is important for infections at mucosal surfaces, IgG would be really important in the third example we have here, where there's direct infection of the bloodstream via needle or bites that then spread to target organs, for example, hepatitis B, rhabdo. So in these cases, the organisms aren't entering the body via a mucosal surface, they're going straight into the bloodstream. Therefore, the IgG will be required for this viral clearance in the blood. So in this slide, we're going to talk a little bit about the polio vaccines. Okay, there's two vaccines, and they're quite important. So first of all, just a little bit of background about polio. So polio is a serious viral infection, and it used to be common in the UK and throughout the world, but it's rare these days because of the vaccination. Okay. Most people with polio don't have any symptoms and won't know they're infected, but for some people the polio virus causes temporary or permanent paralysis, which can be life-threatening. And in the UK, as we said at the beginning of the lecture, case of polio fell dramatically when the routine vaccination was introduced mid-1950s. And there hasn't been a case of polio caught in the UK since the mid-80s. There is no cure for polio, so it's important to make sure that children are fully vaccinated against it. So the first polio vaccine that was developed was the Salk vaccine in 1955, and this was an example of an inactivated vaccine where the virus particles have been treated with formaldehyde so that they can't infect the host cells. Then in 1961, a new polio vaccine was developed, an oral vaccine, which was delivered on sugar cubes. So the oral vaccine, also called OPV, or the Sabin vaccine. So this had the advantage over the Salk vaccine that it was given by mouth rather than injection. And the viruses it contains can spread to the other members of the vaccines of the person who had the vaccine's family, thus immunizing the family as well. However, the disadvantage with this oral vaccine, because it's an example of an attenuated virus, a weakened virus, this has the disadvantage that on the rare occasions, one of the strains in the vaccine regains full virulence and causes the disease. And for this reason, the Salk vaccine has once again become the preferred vaccine worldwide. So people have gone back to the inactivated vaccine. So as we said, the problem with the oral vaccine, the Sabin vaccine, is that it's a live vaccine, and you do get occasions where the vaccine, rare occasions where the vaccine regains full virulence and causes a disease. And you do get around 10 cases of vaccine-associated disease per year. And these cases are called revertants. This is because the virus has reverted back to full virulence. And 1 in 4 million vaccine infections give paralytic polio. So the patient becomes paralyzed. So as we said, for this reason, people have gone back to the inactivated vaccine, the Salk vaccine. But this also has its disadvantages. So immunity is more difficult to establish and boosters are needed. And doesn't stop the gut infection if the wild type virus is circulating in the population. So this slide looks at the recombinant subunit vaccine for Hep B in more detail. So this is shown, the steps are shown in the diagram here, but just to talk you through them. So first of all, the antigen producing gene is isolated from the hepatitis B virus. Then the next step is a plasmid DNA is extracted from a bacterium and is kept with restriction enzyme, forming the plasmid vector. The isolated antigen producing gene is then inserted into the bacterial plasmid vector on forming the recombinant DNA. Then this recombinant DNA containing the target gene, okay, the antigen producing gene, this recombinant DNA is introduced into a yeast cell, forming the recombinant yeast cell. This recombinant yeast cell then multiplies in the fermentation tank and produces the hepatitis B antigens. The final step is the hepatitis B antigens are extracted, purified and bottled and they're ready for vaccination in humans. I looked at DNA vaccines in more detail. And DNA vaccines represent quite a new and promising approach to vaccination. And DNA vaccines are produced by inserting genes for antigens into a recombinant plasmid vaccine. Introduction of the DNA vaccine into a patient leads to uptake of the recombinant plasmid by some of the patient's cells, followed by transcription and translation of antigens and presentation of these antigens to activate adaptive immunity. The expressed antigenic protein would be exposed to the surface of cells and induce cell-mediated cytotoxicity and memory cells. So DNA vaccines have been shown to give a broad immune response and also a long-lasting cytotoxic T lymphocyte response. And also they don't have the risk of active disease associated with them like live attenuated vaccines does. In addition, DNA is easy to make and stable upon storage, which these are very good qualities for vaccines. So you can see why this is an area of promising research. So in summary, vaccines play important roles in developing adaptive immunity against various life-threatening diseases. A number of different methods are used to produce vaccines, and each has its own disadvantages and advantages. Host immunity is key in developing immune responses upon vaccination. IgG and IgA molecules are produced depending on the mode of vaccination. And vaccinations often produce lifelong immunity, although in some cases booster doses are required.